So welcome back, everyone. I hope you're all feeling refreshed after the break, and we'll now resume with our new fellow seminar and hear some more excellent talks. And the first one since morning tea is Professor Georgia Chenevix-Trench, speaking to us on the spectrum of breast cancer susceptibility genes.
Thank you very much, and thank you very much to the Academy, and particularly to my sponsors and referees for electing me to this very prestigious group. I'm going to talk today about breast cancer genetics, mainly because that's what I work on, but also because it's a very good example of what we call a complex genetic trait. We used to call these complex really because we didn't understand the genetics behind them, but in the last 5 or 10 years we've actually gained an enormous understanding not only of breast cancer but a huge number of other human traits that have a genetic basis, both normal traits and disease traits. And so now I think we'd call them complex because indeed the genetics are extremely complex. So the fact that breast cancer and often ovarian cancer clusters together in families was first described by Pierre-Paul Broca in the 18th century. And you can see here a really very nasty pedigree with multiple women affected with breast cancer in their early 40s, in their childbearing years, and also some cases of ovarian cancer. Along with age and sex, family history is the strongest risk factor for breast cancer, but there are certainly also hormonal influences, and much of the increase in the last 50 years or so has been because of changes in reproductive practice, obviously not because of the rapid changes in genetics. However, this is an extreme example, and the burden is very great for these families, but most breast cancer doesn't occur in these kinds of families. It occurs in families with no family history at all, or very modest family history. And so we used to call those cases sporadic, but in fact we know that even those have a genetic basis to them, but a much more complex genetic basis. So breast cancer was actually the first of these complex diseases where the gene responsible was linked to a chromosome by Mary Claire King in 1990, and this was such a landmark event that anybody who was at the Human Genetics meeting that year can remember people literally running from the audience back to the telephones to relay this back to their labs. This was a really phenomenal achievement. And it then took 4 years of really intensive competition to clone this gene, the gene that was responsible, and it was called BRCA1. The sort of experiments that were needed back then took armies of people many years, whereas if you're lucky, you could do the same sort of experiment now in a week by sequencing the entire genome. So, as well as BRCA1, which mainly influences the risk of breast and ovarian cancer, there's another gene called BRCA2, which particularly increases the risk of male breast cancer, and these are what we would now call high penetrance genes. The mutations in them are extremely rare, in most populations occurring at less than 0.1%, although there have been bottlenecks, and they're more common, for example, in the Jewish population. But the risks associated with these rare mutations are very high, so that a woman carrying one of these mutations has approximately a 70% chance of developing breast cancer before the age of 70. However, all this excitement in the 90s took place without any contribution from Australian scientists at all. And that changed because Joe Sambrook, who's also a fellow of the Academy, suggested in 1995 that if we wanted to contribute to this field, which was obviously emerging, we needed to get together, we needed to collaborate, and we needed to collect the resources. To do genetics, you basically need blood samples from people who are alive. And so thanks to what's now called the National Breast Cancer Foundation, and the Kathleen Cunningham Foundation as it was, funded us to establish what we call CONFAB, and which has grown in that time to really the best resource in the world for doing this kind of work. They collect an enormous amount of data, an enormous number of biospecimens, and most remarkably, they make this available to anybody in the world with an ethically approved project. And so you can see from that chart there that we supply literally thousands of samples around the world. I ran CONFAB for many years, and that's run from Melbourne by other people, but I think it's done Australian science an enormous amount of good to develop this sort of research and make it available all over the place. And because of that, I was able to actually enter this field myself. I had access to these families and this material. And my main contribution in this time was to show that apart from these very highly penetrant genes like BRCA1 and BRCA2, There are also genes where the mutations are slightly more common, but they have a more modest effect. They perhaps increase your risk 2- or 3-fold. And the gene I particularly worked on in this time is called ATM. And in this case, we were using a completely different approach. So instead of trying to link a gene to a chromosome, which is what was done with BRCA1, and then identify that gene, we used the candidate gene approach, in which you try to guess, based on the biochemistry and the behavior of this gene, whether it might have a role in breast cancer predisposition. And this is a sort of interesting example and personifies what Beatson described as treasure your exceptions. This is an extremely rare disease. It's actually an autosomal recessive disease, so the mutations are carried from both parents, and the children have quite a complex phenotype that includes defects in the immune system and in movement and an increased risk of diabetes, but they also have an increased risk of leukemias and lymphomas. Well, people noted a long time ago that their mothers who carry a single version of this mutation and their sisters had an increased risk of breast cancer. And so we specifically looked in breast cancer families for mutations in this gene and found that sure enough they occur, they're pretty rare, and they cause this modest increase in breast cancer. And this in some ways has become a prototype for other complex diseases where people started to realize that there would be very highly penetrant mutations, but there would also be mutations of moderate penetrance that we should look for. But what's really exploded the field in the last 10 years or so has been what are called genome-wide association studies. The basic principle behind them is extremely simple. You simply measure genetic variants in 2 populations, both in people with the disease or trait of interest and in controls, and you look at the frequency of that genetic variant in cases versus controls. And because we're looking here at predisposition, the DNA is almost invariably taken from blood samples because they're meant to represent the germline genetics. And you can see in this case that the variant 2 that I've depicted in orange is obviously more common in affected people than unaffected. And so that would suggest that it could be related to their susceptibility. And this whole explosion came about because we developed methods to measure hundreds of thousands of these genetic variants relatively cheaply. Our latest experiment will measure about 400,000 of these for $50. Because we had by then collected enormous resources in which to test them. The other thing that this technique really depends upon is the fact that genetic variants occur within blocks in chromosomes that are co-inherited together. And so you don't need to actually measure the millions of variants in the genome to do this. You just need to measure one of the variants that's within one of these blocks, which will indirectly measure all the others in the block. So you don't need to be measuring the variant that's actually causal for the disease itself. You can just measure one of its correlated variants. And so even before this first genome-wide association study for breast cancer was done, we realized that we would need really very large numbers to validate the effects that we expected to see. We knew that we would be looking for genes of small effect, and so to have sufficient statistical confidence, we would need very large studies. This consortium started with about 30,000 cases and controls, and as you can see, is now up to about 200,000. And the really remarkable thing about what's happened in the last 10 years is that people were so convinced they needed to work together that I think almost every researcher in the world who has access to DNA from breast cancer cases and controls is part of this consortium. So you can imagine that that's been very interesting sociologically, and it's also been extremely successful scientifically. And as a consequence, we've now discovered over 100, I think the latest count is 120 genetic variants that we would call low penetrance variants. So these, in contrast, are much more common. They can be as common as 50%, 10, 20, 30%, but they confer very small increases in risk, more like a 10 or 20% increase in risk. Sometimes people put names to these variants, but that's really just because the genes I've crossed out there are the closest gene, but as I'll point out, they're not necessarily the causal gene. So the rationale for doing these studies probably initially was really to try and understand the genetics of complex traits, and that's been enormously successful. There's now a lot of effort trying to use this information to identify people at risk, to try and target them for more or less screening. A certain sector of the population probably doesn't need mammographic screening, others perhaps need more intensive and earlier screening, and perhaps to put them onto different risk reduction medications. But the thing that I'm most interested in, and my lab is pursuing most at the moment, is to use this information to provide insights into the biology of these diseases and to identify new targets for either prevention or treatment, much of which might be done by repositioning old drugs that are used for other diseases. And the sort of proof of principle that this might be successful comes from two examples. These genome-wide association studies have found that the gene HMG-CoA has a variant in it that's responsible for a very modest change in cholesterol levels. And yet statins, which target that drug, that gene, have been one of the most successful drugs of the last century. And similarly, the estrogen receptor, ESR1, has a variant that's associated with breast cancer, and that's the target of tamoxifen. But you can see from the odds ratio there that these are very modest effects, which is really irrelevant. You could have no genetic variant at all and yet have an enormous potential for drug development. So I'll skip over this, which essentially shows that we did a very large experiment last year that found about 50 new genes, and point out that these genes often occur in what we call gene deserts, a long way from anywhere else, and sometimes in gene forests. So this is what's called a Manhattan plot, with the SNPs along the chromosome on the bottom and the p-value associated with them on the y-axis. And this shows you that there are a lot of different genes down here. This is a variant associated with both breast and ovarian cancer, and there are two very good candidate genes nearby, but it turns out that none of them are actually involved in breast cancer susceptibility. We know that the relevant polymorphisms, genetic variants of these ones here, and my collaborators in Brisbane have recently been able to show that those genes, those variants actually target this gene here, which we knew absolutely nothing about in terms of its breast cancer risk. And we can do that by showing that those genetic variants lie in what are called enhancers that loop in the chromosome to the promoter of that nearby gene. And this just shows you that this looping occurs from the promoter in this gene to this region here, which is an enhancer, and in which we have specific polymorphisms that we know increase risk of both breast and ovarian cancer. We have another example here on chromosome 10 in a region associated with both breast cancer risk and breast density, which is a risk factor. And in this case, we've got two different regions of polymorphisms, region 1 and region 2. But in both cases, we've been able to show that they interact with the promoters of genes nearby, a tumor suppressor gene here and a nuclear receptor gene here. And so what we think is that many of these associations that we find in genome-wide studies act because they exist in enhancers that can target not just one, but multiple genes in the vicinity. And the reason that this is very important is that we know that these genome-wide association study hits, as we call them, are enriched in genes where there's already a drug, but we really need to know what that gene is in order to find opportunities for drug repositioning and use, for example, one of the things I'm working on is a drug developed for sickle cell anemia that we think might be relevant for breast cancer prevention. So I'll just end it there and thank my husband. We are now, I think, the fourth couple in the academy, Nick Martin, my lab, who I believe are listening, and my friends, including our new fellow Craig Moritz here, looking a little younger and browner. Thank you.
Thank you very much, Georgia. Questions? Roger.
Just short, Melbourne University, husband of Marilyn. I'd like to ask you two questions in one. Why is it that nuns have the highest incidence of breast cancer of any community in the world? And secondly, what is the explanation for the fact that if you breastfeed one or more children, that gives you a major protective effect against getting breast cancer in later life?
Well, the simple answer to nuns, as you know, is that it's because they don't have multiple children, they don't breastfeed them a long time, they don't have early children. Conversely, of course, because they multiply ovulate, they increase their risk of ovarian cancer. In terms of breastfeeding, to be honest, I don't know those answers. You probably know them better than I do. It's not really my area of expertise, so I won't make it up.
Eugenie.
Metformin has been suggested as a drug that can be repositioned for breast cancer. Is that for specific genetic variants of breast cancer, or what?
I know that that's the case, and I think there are clinical trials of that, but I don't know that actually the biology behind that has been sorted out yet. I think it's a case where there's a genome-wide association study that predicts that metformin will be effective, but they're actually skipping some of this very intensive work, which I skipped over, and heading for the target itself and seeing if it's effective.
If I could just ask another quick question. In your telangiectasia, is that associated with increased angiogenesis? And is that a biological basis for the mothers having an increased risk of breast cancer?
No, it's heavily involved in recognising DNA damage repair. And one of the real surprises of these genome-wide association studies is that we haven't found a lot of other genes like that, because BRCA1, BRCA2, ATM, PALB2, many of the genes with higher penetrant mutations are involved in DNA repair. We assume the same would be the case for the low penetrant genes. And in fact, that isn't the case, and there's really no class of genes that seem to be targeted. What we often find is that the target gene is a surprise, and even when it's a known gene, the effect is not in the same direction that you would expect. So we can show that the genetic variants that increase the risk of breast cancer are actually associated with a lower level of cyclin D1, for example, and that the genetic variants that are associated with breast cancer are not the ones that influence chromosome length, which we would have predicted. So there's almost all of the loci where we've got a good understanding of the biology, we're finding quite a surprising outcome.
Thank you very much, Georgia, for a fantastic lecture.